Principles of inheritance and variation. Mendel's laws of inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, that is 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. For example, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination or cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, that is, full or constricted green or yellow pods, and tall or dwarf plants. Contrasting traits studied by Mendel in pea. Serial number. Characters. Contrasting traits. Serial number 1. Characters. Stem height. Contrasting traits. Tall or dwarf. Serial number 2. Characters. Flower color. Contrasting traits. Violet or white. Serial number 3. Characters. Flower position. Contrasting traits. Axial or terminal. Serial number 4. Characters. Pod shape. Contrasting traits. Inflated or constricted. Serial number 5. Characters. Pod color. Contrasting traits. Green or yellow. Serial number 6. Characters. Seed shape. Contrasting traits. Round or wrinkled. Serial number 7. Characters. Seed color. Contrasting traits. Yellow or green. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial one progeny or the F1. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf. He made similar observations for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and, to his surprise, found that in the filial 2 generation, some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf was 1/4, that is 25% of the F2 plants, while 3/4, that is 75% of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down, unchanged, from parent to offspring through the gametes over successive generation. He called these things as factors. Now we call them as genes. Therefore, genes are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t for the dwarf. And capital T and small t are alleles of each other. Hence, in plants the pair of alleles for height would be capital T, capital T, capital T, small t, or small t, small t. Mendel also proposed that in a true breeding tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous, capital T and small t respectively. Capital T and small t are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance. He proposed that in a pair of dissimilar factors, one dominates the other, that is, as in the F1, and hence is called the dominant factor, while the other factor is recessive. In this case, capital T, that is, for tallness, is dominate over small t, that is, for dwarfness, that is recessive. He observed identically behavior for all the other characters, that is, or trait pairs that he studied. It is convenient, that is, and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness, that is, do not use T for tall and D for dwarf because you will find it difficult to remember whether capital T and small d are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygotes capital T and small t, or can be dissimilar as in the case of the heterozygote capital T small t. Since the capital T small t plant is heterozygous for genes controlling one character, that is height, it is a monohybrid and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separates or segregate from each other, and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossings. In this way, the gametes of the tall T plants have the allele T, and the gametes of the dwarf t plants have the allele t. During fertilization, the two alleles T from one parent, say through the pollen, and t from the other parent, then through the egg, are united to produce zygotes that have one T allele and one t allele. In other words, the hybrids have T. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett square. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. The Punnett square shows the parental tall T figure, that is male, and dwarf t that is female plants. The gametes understand a typical monohybrid produced by them and the F1 T progeny. The F1 plants of genotype T are self-pollinated. The symbols male and female are used to denote the female, that is eggs, and male, that is pollen, of the F1 generation respectively. The F1 plant of the genotype T, when self-pollinated, produces gametes of the genotype T and t in equal proportion. When fertilization takes place, the pollen grains of genotype T have a 50% chance to pollinate eggs of the genotype T as well as of genotype t. Also, pollen grains of genotype t have a 50% chance of pollinating eggs of genotype T as well as of genotype t. As a result of random fertilization, the resultant zygotes can be of the genotypes T, T, or t. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to T, 1/2 lead to T, and 1/4 to t. Though the F1 have a genotype of T, but the phenotypic character seen is tall. At F2, 3/4 of the plants are tall, where some of them are T, while others are T. Externally, it is not possible to distinguish between the plants with the genotypes T and T. Hence, within the genotypic pair T, only one character T tall is expressed. Hence, the character T or tall is said to dominate over the other allele t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall. That is, though the genotype is T, and in the F2, 3/4 of the plants are tall. That is, though genotopically 1/2 are T and only 1/4 are T. This leads to a phenotypic ratio of 3/4 tall. That is, 1/4 T plus 1/2 T and 1/4 t. That is a 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4 is to 1/2 is to 1/4 ratio of capital T is to capital T is to small t is mathematically condensable to the form of the binomial expression, that is ax plus by whole square, that has the gametes bearing genes capital T or small t in equal frequency of 1/2. The expression is expanded as given below. 1/2 capital T plus 1/2 small t whole square is equal to 1/2 capital T plus 1/2 small t into 1/2 capital T plus 1/2 small t is equal to 1/4 capital T plus 1/2 capital T small t plus 1/4 small t. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarfs was homozygous t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using mathematical probability, by simple looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In typical test cross, an organism, that is, here P plants, showing a dominant phenotype, that is, and whose genotype is to be determined, is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. The results of typical test cross where violet colored flower, that is capital W, is dominate over white color flower, that is small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition for a test cross? Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law or law of segregation. Law of dominance. Number 1. Characters are controlled by discrete units called factors. Number 2. Factors occur in pairs. Number 3. In a dissimilar pair of factors, one member of the pair dominates, that is dominant, the other, that is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, that is snapdragon or antirrhinum species, is a good example to understand incomplete dominance. In a cross between true breeding red flowered, that is capital R, and true breeding white flowered plants, that is small r, as a result the F1, that is capital R, was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1, that is capital R red, is to, that is capital R pink, is to 1, that is small r white. Here, the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1 dominant recessive ratio. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as being from capital R that is red and small r that is white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, that is about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, that is, S is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate, that is capital S. Theoretically, the modified allele could be responsible for production of number 1, the normal or less efficient enzyme, or number 2, a non-functional enzyme, or number 3, no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype or trait, that is, result in the transformation of substrate capital S. Such equivalent allele pairs are very common. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified, that is, functioning allele, which represents the original phenotype, is the dominant allele and the modified allele is generally the recessive allele. Hence, in the above example, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage, though the parents contain two alleles during gamete formation. The factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Dominance. Till now, we were discussing crosses where the F1 resembled either of the two parents, that is dominance, or was in between, that is incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene capital I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene, that is capital I, has three alleles, IA, IB, and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the 3I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and small I are present only, IB expresses, that is because small I does not produce any sugar. And when IB and small I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are 3 different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood types. How many phenotypes are possible? Table showing the genetic basis of blood groups in human population. Allele from parent 1. Allele from parent 2. Genotype of offspring. Blood types of offspring. Allele from parent 1 IA. Allele from parent 2 IA. Genotypes of offspring IA. Blood types of offspring A. Allele from parent 1 IA. Allele from parent 2 IB. Genotype of offspring IB. Blood types of offspring AB. Allele from parent 1 IA. Allele from parent 2 small I. Genotype of offspring IA small I. Blood types of offspring A. Allele from parent 1 IB. Allele from parent 2 IA. Genotypes of offspring IB. Blood types of offspring AB. Allele from parent 1 IB. Allele from parent 2 IB. Genotype of offspring IB. Blood types of offspring B. Allele from parent 1 IB. Allele from parent 2 small I. Genotype of offspring IB small I. Blood types of offspring B. Allele from parent 1 small I. Allele from parent 2 small I. Genotype of offspring small I. Blood types of offspring O. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, that is 3 alleles governing the same character. Since an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, that is, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce small starch grains. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be the dominant allele. But the starch grains produced are of intermediate size in capital B seeds. So if starch grain size is considered as the phenotype, then from this angle, the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. Inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape, and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parent had yellow colored and round shaped seeds. Here can you tell which of the characters in the pairs yellow or green color and round or wrinkled shape was dominant? Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeds plants and between round and wrinkled seeds plants. The genotypic symbols, capital Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital RR YY and small rr YY. The cross between the two plants can be written down as in figure, in which showing the genotypes of the parent plants. The gametes capital RY and small ry unit on fertilization to produce the F1 hybrid capital R small r, capital Y small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green colors segregate in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. Theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy, that is, as it is now, in those days, and his work could not be widely publicized. Secondly, his concept of genes, that is, or factors, in Mendel's words, as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other, was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors, that is genes, were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists, that is, De Vries, Correns, and von Schermach, independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, that is, colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws, the behavior of chromosomes during meiosis, that is, equation division, and during meiosis, that is, reduced division, can be understood and like genes. Chromosomes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. Table: a comparison between the behavior of chromosomes and genes. A. Occurred in pairs. B. Occurred in pairs. A. Segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. B. Segregate at gamete formation and only one of each pair is transmitted to a gamete. A. Independent pairs segregate independently of each other. B. One pair segregates independently of another pair. Can you tell which of these columns, A or B, represents the chromosomes and which represents the gene? How did you decide? During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different colors in the left and right columns. In the left column, that is possibility 1, orange and green is segregating together. But in the right hand column, that is possibility 2, the orange chromosome is segregating with the red chromosomes. Possibility 1, one long orange and short green chromosome and long yellow and short red chromosome at the same pole. Possibility 2, one long orange and short red chromosome and long yellow and short green chromosome at the same pole. Sutton and Bovary argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexually reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny files. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. Topic 8. Law of independent assortment. In the dihybrid cross, the phenotypes round yellow, round green, and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow:1 green with 3 round:1 wrinkled. This derivation can be written as follows. That is, 3 round:1 wrinkled, 3 yellow:1 green is equal to 9 round yellow:3 wrinkled yellow is to 3 round green is to 1 wrinkled green. Based upon such observation on dihybrid crosses, that is, crosses between plants differing in 2 traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when 2 pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the 2 pairs of genes during meiosis and the production of eggs and pollen in the F1 capital R small r capital Y small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of the small r bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes, that is, 4 types of pollen and 4 types of eggs. The 4 types capital RY, capital R small r capital Y and small ry, each with a frequency of 20% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the 2 sides of a Punnett square, it is very easy to derive the composition of the zygotes that give rise to the F2 plants. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the 2 genes did not segregate independently of each other, and the F2 ratio divided very significantly from the 9:3:3:1 ratio that is accepted when the 2 genes are independent. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the 2 genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the 2 genes and coined the term linkage to describe this physical association of genes on a chromosome and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, that is, showed very low recombination, while others were loosely linked, that is, showed higher recombination. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Stuart Wendt used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequence of whole genomes, as was done in the case of the Human Genome Sequence project, described later. Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. However, there are instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways, which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that code for the enzyme phenylalanine hydroxylase, that is, single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. Inheritance. Mendel's studies mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume that 3 genes A, B, C control skin color in human, with the dominant forms A, B and C responsible for dark skin color and the recessive forms small a, small b and small c for light skin color. The genotype with all the dominant alleles, that is capital AA, BB, CC, will have the darkest skin color, and that with all the recessive alleles, that is small AA, BB, CC, will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. In humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes is present in the female, whereas the presence of X and Y chromosome are determinant of the male characteristics. During spermatogenesis among males, 2 types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female, that is XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. Determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from the union of a sperm and an egg develops as a female, that is, queen or worker, and an unfertilized egg develops as a male, that is, drone, by means of parthenogenesis. This means that the males have half the number of chromosomes than that of a female. The females are diploid having 32 chromosomes and males are haploid, that is having 16 chromosomes. This is called as haplodiploid sex determination system and has special characteristic features such as the male produce sperms by meiosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic or chromosomal mechanisms of sex determination can be traced back to some of the experiments carried out on insects. In fact, the cytological observation made in a number of insects led to the development of the concept of the genetic or chromosomal basis of sex determination. Hanking, that is, 1891, could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to this structure as the X-body, but he could not explain its significance. Further investigations by other scientists led to the conclusion that the X-body of Hanking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the XO type, that is, all eggs bear an additional X chromosome besides the other chromosomes, that is, autosomes. On the other hand, some of the sperm bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperm that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female is equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named autosomes. Grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. This observation led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals including man, XY type of sex determination is seen where both male and female have the same number of the chromosomes. Among the males an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. However, females have a pair of X chromosomes. Both males and females bear the same number of autosomes. Hence, the males have autosomes plus XY, while females have autosomes plus XX. For example, in human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosome besides autosomes. In the above description, you have studied about two types of sex determining mechanisms, that is, XO type and XY type. But in both cases, males produce two different types of gametes, A, either with or without X chromosomes, or B, some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanisms are designated to be example of male heterogamety. In some other organisms, for example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is the same in both males and females, but two different types of gametes in terms of the sex chromosomes are produced by females, that is, female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In these organisms, the females have one Z and one W chromosome, whereas males have a pair of Z chromosomes besides the autosomes. The mutation is a phenomenon which results in the alternation of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, the mutation is another phenomenon that leads to variation in DNA. As you will learn in the next chapter, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, that is deletions, or gain, that is insertion or duplication, of a segment of DNA results in alteration in chromosomes. Since genes are known to be located on chromosomes, alternation in chromosomes results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, the mutation also arises due to a change in a single base pair of DNA. This is known as a point mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA cause frame shift mutation. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutation. These are referred to as mutagens. UV radiations can cause mutations in organisms. It is a mutagen. Disorders. Pedigree analysis. The idea that disorders are inherited has been prevailing in human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance patterns of traits in human beings began. Since it is evident that control crosses that can be performed in pea plants or some other organisms are not possible in the case of human beings, the study of the family history about the inheritance of a particular trait provides an alternative. Such an analysis of traits in several of generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, the pedigree study provides a strong tool, which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alterations do take place occasionally. Such an alteration or change in the genetic material is referred to as a mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. It is a sex-linked recessive disorder due to a defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female, that is carrier, for hemophilia may transmit the disease to sons. The possibility of a female becoming hemophilic is extremely rare because the mother of such a female has to be at least carrier and the father should be hemophilic, that is unviable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carriers of the gene, that is, or heterozygous. The disease is controlled by a single pair of alleles, HBA and HBS. Out of the three possible genotypes, only homozygous individual for HBS, that is HBS-HBS, show the diseased phenotype. Heterozygous, that is HBA-HBS, individuals appear apparently unaffected, but they are carriers of the disease as there is a 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid, that is GLU, for valine, that is VAL, at the 6th position of the beta-globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta-globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from a biconcave disc to an elongated sickle-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of this in the brain results in mental retardation. These are also excreted through urine because of its poor absorption by the kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the parents are the unaffected carriers of the gene, that is, or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in a reduced rate of synthesis of one of the globin chains, that is, alpha and beta chains, that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules resulting in anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, the production of alpha globin chain is affected, while in beta thalassemia, the production of beta globin chain is affected. Thalassemia is controlled by two closely linked genes, HBA1 and HBA2, on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the fewer alpha globin molecules produced. While beta thalassemia is controlled by a single gene, HBB, on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. Disorders. The chromosomal disorders, on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatides during the cell division cycle results in the gain or loss of a chromosomes, called aneuploidy. For example, Down's syndrome results in the gain of an extra copy of chromosome 21. Similarly, Turner's syndrome results due to the loss of an X chromosome in human females. Failure of cytokinesis after the telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 46, that is 23 pairs. Out of these 22 pairs are autosomes and 1 pair of chromosomes are sex chromosome. Sometimes, through rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome respectively. Such a situation leads to very serious consequences for the individual. Down's syndrome, Turner syndrome, and Klinefelter's syndrome are common examples of chromosomal disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same line as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in figure for dominant and recessive traits. Discuss with your teacher and design pedigrees for characters linked to both automos and sex chromosome. Down's syndrome. The cause of this genetic disorder is the presence of an additional copy of chromosome number 21, that is, trisomy of 21. This disorder was first described by Langdon Down, that is, 1866. The affected individual is short-statured with a small round head, furrowed tongue and partially open mouth. The palm is broad with a characteristic palm crease. Physical, psychomotor, and mental development is retarded. Klinefelter's syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a cryotype of 47 XXY. Such an individual has overall masculine development. However, the feminine development, that is, development of breast, that is, gynecomastia, is also expressed. Such individuals are sterile. Turner's syndrome. Such a disorder is caused due to the absence of one of the X chromosomes, that is, 45 with XO. Such females are sterile as ovaries are rudimentary besides other features, including lack of other secondary sexual characters.